Yeah, as I've just been nicely introduced, I have actually just started. It's been only 3 months, and I use both computational methods and then a little bit of wet lab biology to understand how we educate our immune system. And while I don't expect all of you to know how the immune system works in detail, I'm sure most of you have heard about instances where the immune system actually goes rogue. You might suffer yourself, or like you might know people that suffer from allergies, where the immune system reacts too strongly against something that is actually not very harmful to us. Or on the other end of the spectrum, autoimmune diseases such as type 1 diabetes, multiple sclerosis, Crohn's disease, where the immune system mistakes the cells from our own body as foreign and starts attacking them, which ultimately leads to their destruction. And while I think it's very important to understand and study autoimmune diseases, I think it's equally important to understand how the healthy processes work in the immune system. And if we know in detail how the healthy mechanisms of educating the immune systems work, then we might be able to pinpoint down where things go wrong and take these insights to ultimately then combat autoimmune diseases. And we have very many different cells in our body that act as this sort of police force in our body to find cells that are infected with foreign pathogens such as bacteria or viruses. And I will focus on one cell type specifically. They're called the T cells. And I've indicated them here in like little gray dots. And T cells come from the bone marrow. And once they leave the bone marrow, the T cells get educated. So how do the T cells know which parts of our body are healthy and which parts are infected by some sort of pathogen? And the T cells have devised a system where they say, instead of trying to learn everything foreign that I could encounter, I'm going to learn everything that belongs to our own body, which is a huge task, as you can imagine. If you think about the tissues in our body and how specialized they are, you know, your eye cells basically have proteins that can capture light. And by this mechanism you can see. Or we have proteins in our muscles that can contract and make us move. So somehow the T cell has to learn all of these different proteins in the different tissues. Sort of like students that know nothing. They're like just like before they go to the first day in school, and now they have to go to the school or the place where they can learn this, and they go to an organ, the thymus. And the thymus sits atop of our heart. It's very active in many of you, mostly kids that haven't got into puberty yet. And by the age of 50, it's mostly inactive. So the development of our immune system is really mostly active while we're young. And in the thymus, these T cells, little students, spend a few days where they try to learn all the proteins that they could encounter in the body. And once they leave it, in a slightly different conformation, here just indicated with another gray dot, they're then fully mature, and when they see a foreign invader, they can then become active and fulfill their task as a sort of policeman. I'd like to think of these cells as if they have graduated, they know it all, but in order to understand what happens from tiny little students to graduated students, it's helpful to understand a little bit better how the thymus basically works. So I've drawn a little cartoon here of the thymus. Our little T cells are here in gray, or we follow like one T cell here in purple. And the T cell comes into the thymus, but it doesn't just sit at one spot. Sort of migrates around the thymus, and you can see it attaches to different cells. So it's actually interacting with the cells in the thymus. So these thymus cells are actually really important. They're the crucial bit in the educating of this part of the immune system. And as you can see here in tiny wiggles, these cells express different kinds of proteins. So the cells in the thymus tell the T cells what is part of our own body and what isn't. And I've told you before, we have all these different proteins in our eyes, in our muscles, nose and liver. So somehow the thymus has to represent all of these proteins. And I think a good analogy is thinking of the thymus as a library, where all the cells in the thymus are books in the library, and all these different proteins are words in these books. And the T cell's task is now to go into the thymus and learn all the words in all the books in the library. And it's quite a daunting, it's quite a challenge really. And I thought we could have a little interactive task to show you how difficult, or maybe that it's actually less difficult than we might think. We'll find out. So I would need 4 volunteers that are happy to read a single sentence out of the book. Yes, I found one already. I'm just going to bring you the books. Do I have 3 more volunteers? Yeah, come on up. 2 more? Yes. Okay. We just read the sentence. Once your turn, this one, the other sentence. And then we have this one. And I need 1 more, if possible. Somebody? If not, I will read it myself. Okay. Hello. Okay, so basically we have 4 seemingly unrelated books. We have a travel guide, we have a novel, we have a somewhat technical book, and then we've got a cookbook here. What's your name? Mark is going to read the first sentence out of the travel book. Do you want the microphone?
I'll take it. Although I think everybody can hear me without it. Is that true?
You want to try though?
Okay. I like microphones. In summer, there are scores of free events around town.
Okay, perfect. What's your name?
Michael,
Okay, Michael's going to read the next sentence.
at this point, I should explain what had happened to me since that summer day.
Let's see, what's your name?
Like,
Michael, look.
have a look at a simple puzzle.
Perfect. Okay, and then I'm going to read the last one out of the cookbook. This dish really reminds me of summer. Thanks, guys. Thank you. Okay, so 4 seemingly unrelated books, 4 somewhat random seeming like sentences that seem very random, but if you look at them, if we go through the first sentence, in summer there are scores of free events around town, you can actually see that the word summer was both in the travel guide, in the novel, and then in the cookbook as well. And we can do this game a little more, and we can go through the other words in these sentences, and we can see the little word off occurs a few times. Actually, the period at the end of the sentence, we find 4 times. We can do this a little more, and we find like another few words that occur actually in all of these sentences. So if you imagine we were a little T cell and we have to learn all of this, actually we might not have to read all the books to learn all the words. And that's basically the problem that I'm interested in. How many cells does a T cell have to see in the thymus? How many cells does it have to interact with? How many books do you have to read to learn all the words? And I do this by mostly staring at the screen like this. So this is literally what it looks like. It's mostly a blank screen with some writing on it. And I use a lot of, well, it's all done on the computer, and then I use mathematical models. So ideas about what do the cells represent, how far would a T cell migrate, how much time does it spend there, how many interactions. And then I can build hypotheses about what would happen. And then I can go back to the lab, to the actual wet lab, and try some of these hypotheses to see if these hold true. And this is one example that I've been working on before I started working here. So I've done some simulations, and what this is just representing is all the cells in the thymus, the books in the library, I just simulate as a bag of cells. I know nothing about how the T cell walks through the thymus, how many cells it actually sees. So all I can do is being like, okay, I simulate this by saying I just grab in the bag and pull a cell out at a time and try to understand what's happening in the cell. And then very many of my results would look like this typically. It'll just be some sort of plot, and everybody always wants to see the plots, and we've got some data. And I'll just tell you very briefly what this actually means. So we would always describe something like, what do we display on the x-axis, which is something that we want to study. And on the y-axis we have the outcome. And I've studied different types of interactions. So I'm basically saying the T cell came in and saw 500 cells in the thymus. So the student read 500 books. And if I then say, well, was a good student, he actually read all the words in the book, he gets a 1, but it was a poor student, he skipped a lot of words, then we actually only have like 20% of all the words that are covered. And then I can look at the antigen coverage. So after I met 500 cells, how many possible words have I seen? And you can see if the student was lazy, actually this is quite low. But then I don't actually need to have seen all the antigens. I don't need to be that good. It's already good enough to read about 80 or even only 60%. And that's one little indication, but again, it's only just a bank of cells. And what I'm working on at the moment, which is the non-computational part, I'm trying to understand how this migration pattern works, and I'm looking in mouse models to understand this better. We can use proteins that are tagged with a fluorescent protein that we can follow. And I'm hoping once I've done this, instead of simulating the thymus as a bag of cells, I can actually simulate it in a spatial pattern and can then also understand the interactions much better. And what I hope this will tell us is the healthy processes that happen in the thymus, and then as I indicated before, we might then be able to look at diseased patients and see what's going wrong and then get indications of what we have to do next. Yep, that's what I do.